In today's video, I'm going to talk about vaccine types and how vaccine works. Before we talk about vaccine, let's take a look at virus structures. Let's take the influenza virus as an example. The virus is made up of two parts, the nucleic acid and the protein envelope. There are many types of epitopes on the protein envelope, which determines the virus function and virulence. And then let's look at how virus is killed by our immune system. Basically, there are two types of pathways, MHC class 1 pathway and MHC class 2 pathway. MHC class 1 and MHC class 2 is the name of molecules that involve our self-immune system. So let's take a look at MHC class 1 pathway. So a virus infects the cell and then use the infected cell's organelles to do self-replicates, which will produce virus protein. And the virus proteins bound by MHC class 1 molecules in ER. And then MHC class 1 molecules transfer the virus protein onto the cell surface. And then the MHC class 1 molecule with virus protein binding to it will be recognized by the T cell receptor on the cytotoxic T cell. As soon as the molecule is recognized, the cytotoxic T cell is being activated, and then it will release perforins and granzymes, which will kill the infected cells, because the perforins and granzymes will cause target cell lysis. And MHC class 2 pathway. So first, the macrophage or B cell engulfs and degrades the bacterium or virus, producing peptides. So the peptides is originally part of the antigens on the bacterium or virus. And then the bacteria or virus peptide bind to the MHC class 2 in vesicles. And then MHC class 2 molecule transfer the virus peptide onto the cell surface. And then the MHC class 2 molecule with virus peptide bind to it, recognized by the T cell receptor on the T helper cell. And the T helper cell will release the cytokines that will further activate the macrophage or B cells to let the cell engulf more virus or bacteria. And also the cytokines will cause inflammation and activate other immune cells such as B cells and T cells, and tell the immune system to grow more immune cells like B cell, T cells, and cause the T cell, B cell to differentiate into different types and facilitate antibody synthesis. So let's review what is antibodies and how it works. When the virus or bacteria first infect our cell, the B cell recognize the antigen on bacterium or virus, engulf them into the B cell, and the B cell will degrade the degraded the bacterium or virus, and the virus peptide will bind to the MHC class 2 molecule, and the MHC class 2 molecule transfer the virus peptide onto the B cell surface, and then MHC class 2 molecule will be recognized by the T cell receptor on the T helper cells. And the T helper cell will release the cytokines, will further activate the B cell to engulf more virus, and also help the B cell to develop into the plasma cells and memory B cells. The plasma cell will produce antibodies. And during the second infections of the same virus, during the second infection will cause the memory B cells to develop into plasma cells more quickly and produce a larger amount of antibodies. And how antibody works. So basically, antibodies will bind specifically to the epitopes on the virus. For example, the antibodies which can bind to the measles virus will not bind to influenza virus. And antibodies protect our body by binding specifically around the target virus. And when the virus is fully bounded by the antibodies, the virus will no longer be that easy to infect human cells or animal cells, because antibodies being binded, it cannot be recognized by the receptors on the health cells, so it will no longer be easy to enter our health cells and to produce their own proteins to make the next generation virus. So the fully bounded virus will then recognized by the macrophage of B cell. The macrophage of B cell will engulf the virus and then go through the MHC class 2 pathway and help our body to produce more antibodies, and the virus itself will be degraded by the macrophage or B cell. And so vaccine is killed or inactivated virus that will be introduced into our body to let our body to develop more antibodies, but no longer cause disease. So during the second infection of the real virus, we already have the antibodies developed by the vaccine, so we'll be no longer infected by the same virus. And the very first vaccine was invented by Edward Jenner. So Edward Jenner found that the patient who was previously infected by smallpox will no longer be infected by smallpox. So the theory is that the smallpox virus and smallpox share some epitopes. For example, in this diagram, the epitopes on the smallpox, like the blue one and green one, is also on the smallpox virus, which means when we develop the antibodies specific to the smallpox, especially the antibodies specific bind to the green epitopes and blue epitopes will also be able to bind the green and blue epitopes on the smallpox. So the immunity developed by the chorpox infection will naturally convert to smallpox infection. There are basically two main types of artificial vaccine: the live attenuated vaccine and acute vaccine. So the live attenuated vaccine is not as toxic as the pathogenic vaccine. So how we develop this kind of vaccine, and that we isolate the pathogenic vaccine from patient and grow pathogenic virus from the patient and grow those virus in human culture cells. And then we will get a large amount of pathogenic virus. And then we transfer those virus into monkey cell cultures. So the virus which is grow really well in human cells will not grow well in monkey cells. But in order for the virus to grow well in monkey cells, the virus acquires many mutations that allows it to grow well in monkey cells. And after a few generations of the virus will no longer grow well in human cells. So it is not as virulent as the original pathogenic cells, which said it is being attenuated. And there are other techniques that we can produce live attenuated vaccine is by using DNA recombinant method. So we just remove the virulence part of the DNA and recombine the other DNA part together and transfer this modified DNA to the virus. So the mutate virus gene or delayed virulence gene into the virus. These two kind of virus will not as toxic as the original virus, but it is still alive, it is still viable, which means the virus can still infect the health tissues, but no longer be that toxic. The live attenuated vaccine will go through both MHC class 1 pathway and MHC class 2 pathway. The other type of vaccine is called inactive vaccine. So basically, we have an intact virus. We use a low energetic electron radiation to cause virus death. And we use this inactive virus as a vaccine to be introduced in our body tissues. This kind of vaccine will only go through the MHC class 2 pathway, which will cause our human body to produce antibodies. But this kind of vaccine will not go through the MHC class 1 pathway because since the virus is dead, it can no longer infect the health body tissues. So it will not get into the health tissue cells to produce its own proteins by using the cell organelles. So it will no longer be able to go through MHC class 1 pathways. So basically, this is vaccine and how the vaccine is developed and worked.